So, very good morning, good afternoon, good evening, everyone. So, we are going to discuss about in this video about electrophysiology of the heart. So, electrophysiology of the heart. So, particularly, just take basic introductions about the heart. You all knows in your human anatomy and physiology classes, we have already covered what actually the different parts of the hearts and how it works. So it's just an overview. There are a left atrium, left ventricles. There is a right atrium, right ventricles, and these left atrium, left ventricles, particularly left ventricles, push the blood to the aorta, and then blood goes to the arteries. Then it goes to the capillaries, where exchange of oxygen and nutrients are happen, and then your blood return back to your right atrium, right ventricles, and then it goes to the lungs. This blood is your deoxygenated blood, and in the lungs there is a exchange of your oxygens and carbon dioxide. So your deoxygenated bloods uh, become oxygenated blood, and then it will be again. This is your cardiac cycle, right? So, what actually, how these ventricles contract, what are the different phases that we are going to discuss, and that we call electrophysiology of the hearts. So, we have discussed earlier, about this, but you can see in pictures, you will get better clarifications about these. So there are different phases when we talk about electrophysiology of the heart. So you can see that there is a action potentials. Every cells have a action potential, and that potentials at resting phase, when there is a no stimulus, that resting potential we called RMP, resting membrane potential. So resting membrane potential of this cardiac cell is minus 90 millivolt. So, whereas other cells resting membrane potential is minus 70 millivolt. So question is why these resting membrane potentials are in negative side, even though it's at the resting phase. So that means there are some things which is inside the cells is providing the negativities to the cells, to the potential. So what are these? These can be ions, these can be DNA, which is made up of your phosphodiesterase linkage in the DNA. So all these are the contributors for the negative ions, and so that's why your resting membrane potential is in negative, and that is when we talk about cardiac cell, it's minus 90 millivolt. So you can see that this minus 90 millivolt, whenever there is stimulus, so what happened first? So there is a movement, the ion channels opens, and particularly positive sodium, sodium channels are opened. And whenever sodium channel opens, so you can see that this is cell membrane, this is the outside the cells that we call extracellular fluid, and this is environment inside the cells that we call intracellular fluid. So whenever sodium channel opens, so sodium will move from outside to inside. And why it is so? The reason is because concentration of sodium outside the cell is more, and inside the cell it's less. So obviously there is a movement of the ions from higher concentration to lower concentrations. So that's why whenever sodium channel open, the movement of ions will happen from extracellular fluid to intracellular fluid. Or you can say that the movement of ions happens from outside the cells to inside the cells. So when these ions, sodium ions enter inside the cells from outside, whenever there any stimulus, so what will happens? Your action potential, resting membrane action potential is your negative, so it will goes towards the positive sides. So when it reach at the threshold value, so voltage gated sensitive sodium channel will open, as its name indicate. These are the channels. These channels will open only when your voltage is, or when your action potential reach up to threshold value. Otherwise, these channels will not open. That's why we call it voltage gated sensitive sodium channels or calcium channels. So what will happen when these channels will open? So your action potentials will goes towards positive sides. It crosses the zero and it goes towards the positive sides, and that makes one phase, and that phase we call phase zero. So in phase zero, what's happening? You can see that phase zero, there is a fast sodium channel open, causing fast inward current. upstroke and as a sodium channel rapidly inactivity. So at this phase, at the end of the phase zero, what's happening? At the end of phase zero, so your sodium channels which are opened during this phase, it's going to close. So phase zero is started when your when your sodium channels are open, and when your sodium channel closed at the end, your phase, that is the end of your phase zero, right? So this is phase zero. Then there is a phase one, two, three, as well as four as well. So what happens in other phases? So when we talk about phase one, so in phase one, what's happening? So you can see in these pictures. So now phase zero, at the end of phase zero, your sodium channels are closed. So now sodium is there, but sodium is not moving from outside to inside. And why it's not moving from outside to inside? Because the channels which helps in the movement of sodium from outside to inside, it's closed. So sodium is not able to enter from outside to inside. Then another things which is happening in the phase one is there is the opening of potassium channels. So now you can see that potassium channels are open, but potassium is moving from inside to outside. Now question is why potassium is moving from inside to outside, whereas sodium is moving from in earlier phase from outside to inside. The reason or to answer to this question is potassium, whenever potassium channel open, the movement of potassium is always happen from intracellular to extracellular. And answer why it's happening. The reason is because potassium's in normal conditions, in normal state, your potassium's inside the cell is more, whereas potassium outside the cell is less. And as told you, or every one of you knows that the movement of ions will happen from higher to lower concentrations. So in normal state, potassium concentrations inside the cell is high, outside it's less. So whenever potassium opens, so potassium will move from inside to outside. Whereas there is a vice versa case with sodium. Sodium is outside the cell is more and inside the cell it's less. So whenever sodium channel open, they are moving from outside to inside. And whenever potassium channel opens, they are moving from inside to outside. So, and due to this reason, even though potassium has a positive charge, but there is a decrease in the action potentials. Your action potential is going towards lower side. And why it's happening? Because ultimately your cell is losing the cations. Your cells are losing the positive charge because positive charge potassium channels are moving from inside to outside. So potassium's your positive charge is going to be loose, so your action potential is going to drop. So you can see that in phase one, there is a drop in the action potential. And why it's happening? Because sodium channels are closed and potassium channel are open, and potassium is moving from inside to outside. So ultimately the cells is becomes less positive. So there is a decrease in the action potentials. So you can see that phase one, there is a decrease in action potential. Now, talking about phase two. So in phase two, what's happening? So this is next scenario in the phase two. Phase two we also called as a plateau phase. So you have to see. I hope you might observe one thing. Potassium is opening from here, and potassium is remain open up to phase three. So phase one, phase two, phase three, potassium will remain open. Other science movements will change, but potassium will remain open up to phase three. So now phase two, again potassium is open, but there is a calcium channels which are also open. So potassium is moving from inside to outside, whereas calcium is moving from outside to inside. And again, reason are same because of their concentration differences, because of their concentration gradient. Means outside the cells, calcium's concentration is high, inside the cell it's less. So whenever calcium channel open, the movement will happen from outside to inside. And when calcium is moving from outside to inside and potassium is moving from inside to outside, so for the smaller period of time, this action potential will remain stable. And why it's remain stable or it becomes a plateau? The reason is because one is, cell is losing cation as well as cell are getting the cation as well. So for very small period of times, your action potential will remain stable, and that's why we call it a plateau phase as well. Because sir, there is a calcium is moving. uh, Question can be arise in your mind, calcium have a two positive charge, so why not so why not action potential is going towards positive sides, whereas potassium has a single charge. The reason is because influx of calcium is slow from outside to inside, so it's balancing the these two things, and your action potential will remain stable for a smaller period of time, and that makes another phase, and that phase is phase two, or we also call it a plateau phase. Then next phase is phase three. And I told you one thing, whether it is phase one, two, or three, your potassiums will remain open. So if potassium is remain open, so cell is losing the cations because potassium is moving from inside to outside. So action potential will drop. So at the end of phase two, what happens? Your calcium is going to close. So calcium is closed, but potassium remained open. So calcium is not moving from outside to inside, but potassium is still moving from inside to outside the cells. So ultimately what's happening? Your cell is losing the cations. So your action potential is going to decrease. So that makes your phase 3. So in phase 3, at the end of phase 2, your calcium channel is closed and potassium is remained open. So potassium is remained open, so there is no ion channel which will balance it. So it's going to decrease. And that makes the that phase we call phase 3. And then phase 4. Phase 4 is, I hope you are able to see it. Phase 4 is, we also called forward current. It again means if it increasing the sodium permeabilities, it increase the depolarizations, and next action potential fire as a threshold value reach. So this is makes the phase 4, and whenever your action potential reach up to threshold value, you can see here, this is your threshold value. So there is a starting of phase 0. So there is a opening of your voltage gated sodium channels, voltage gated sensitive channels, so that will make the action potential towards the positive side. So these are the different phases when we talk about electrophysiology of the heart. So again, I can show you. I hope it's clear. So when we talk about electrophysiology of the heart, so first you have to understand what are the different anatomy. I mean, what is the different parts of your heart, how it works. So all these things you have to know, and how your ventricles are contracting. So in ventricles there is a Purkinje's fibers. So how it started. So quickly phase 0, sodium channel is open. So it makes the phase 0 when your action potential reach up to threshold values, then there is a voltage gated sodium channels are open. This that makes the phase 0. And in phase 1, what's happening? Your sodium channels are closed, but potassiums are open. So there is a little bit decrease in the action potentials. But in phase 2, there is a stable phase or plateau phase for smaller period of time. And what's happening in this phase 2? Your potassium is remained open, but your calcium is open, and calcium is moving from outside to inside. So it balance for a very small period of time. And then there is a formations of phase 3. And what happened in the phase 3? Your calcium is closed, but potassium is remained open. So potassium is moving from inside to outside, and there is a decrease in action potential. So this is your electrophysiology of the heart. So now you can understand that means what happening, how these ions are moving. So what's happening in these movement, your sodium inside the cell is increased. In phase 0, your lot of sodiums comes in. So how this balance, means how this is corrected, this ionic balance, because in normal conditions inside the cells your sodiums are less, and and your potassium ions are more inside the cells in normal conditions. So how its means balance is maintained? There is a sodium potassium channels on cell membrane. So on each and every cell membrane there is a one very important channels that we call sodium potassium channels. So this is your sodium potassium channels, and these channels you can see the movement of ions. This there is a movement of 3 sodium from inside to outside, and 2 potassium is coming from outside to inside. This part is outside the cells, this part is inside the cells. So 3 sodium is going from inside to outside, and 2 potassium is coming from outside to inside. And again, so this particularly, I told you, normal conditions, sodium outside the cell is more, and inside the cell sodium is less. So you can see that the movement of these ions are happening against the concentration gradient. Against the concentration gradient means the ions are moving from low concentration to higher concentrations, because outside the cell sodium is already more, and inside the cells your sodium is less. So even though your sodium is moving from inside to outside, so this is a movement against the concentration gradient, and that's why to run these channels you need the energy. And you might we have already discussed about primary active transport, secondary active transport. So this is a classical example of primary active transport, where energy is utilized for the movement of ions across the concentration gradient. So inside the cells already potassium is more, and you see the movement. So movement is happening against the concentration gradient, right? So how these happening, because whenever there is a ventricular contraction, so a lot of calcium comes in, sodium comes in. So all these ion channels which are present on the cell membranes will correct this balance. So how these correct? For example, so, so calcium comes in, sodium comes in, and it contracts the ventricles. So it eject the blood to the aorta, then arteries, then capillaries. So after that, your cardiac cells has to be back to its resting phase. So how it's happening? So whatever the calcium comes, the extra calcium will stored in the smooth endoplasmic reticulums. If still there is a calcium, so it will be moved from inside to outside by one exchanger, and that exchanger is sodium calcium exchanger. So calcium will go from inside to outside, but what's happening? Sodium is coming in with this exchanger. So you can correct the calcium. You can decrease the calcium levels after the contraction, because now you want relaxations. So your calciums will be moved by these channels from inside to outside. But as I said, but what's happening? Your sodium is also coming in. So now next you want to move the sodium from inside to outside. So which how sodium is moving from inside to outside? This is the sodium potassium channel is helping, and sodium is going out and potassium is coming in, and your cells becomes at the comes to the resting phase. And in the resting phase, as we many times we have discussed, the sodium inside the cell is less and potassium inside the cell is high, and vice versa outside the cells. So it will correct in that way. So sodium potassium ATP is formed. What it do, it correct ionic changes of depolarizations and repolarizations, maintain resting potentials, occurs in many isoforms in different tissues. All isoform can be affected by the drugs, that we will discuss in upcoming videos, how drugs can affect these channels and how we can use it. But you can understand that how your ventricles are contracting, there is a movement of cations from outside to insides, and then this is we have discussed phase 2. So ventricle contraction is dependent on calcium ions. So calcium triggers smooth endoplasmic reticulum, contains the receptors for calcium channels, calcium ions, and the endoplasmic reticulum, smooth endoplasmic reticulum is storehouse of your calcium ion. So calcium will move, calcium will bind with proteins, tropomycins, and finally actin and myosins interact with each others and result in contraction of ventricles. And as I said, after contraction of ventricle, it has to be relaxed. So how it's relaxing? Your calcium is storing inside smooth endoplasmic reticulum. Some calcium is going to be exchanged with sodium calcium exchanger, and then there is a sodium potassium ATPase. This is doing the movement of sodium and potassium. So this is all about your electrophysiology of the heart, how are the different phases are formed, how contractions are happening. In next upcoming videos, we will discuss about CHF, that is a congestive heart failure. First, we will discuss it in detail about pathophysiology of congestive heart failures, and then we will discuss pharmacology, how that pathophysiological conditions can be corrected by the pharmacological drugs. So this is all about. Thank you.